Hey everybody, it's Matt Powers, author, educator, seed saver, farmer, citizen scientist, and family guy. And I wanted to tell you about this one because it's something that I created. It's something that I'm sharing with the people I train, but it's also something that you can easily understand. It's something that we're just going to dive into right now. Here we go. So we're looking at here a side by side. And this literally is different samples that we're viewing side by side. That's exactly what it is. And so you can compare in real time the difference between things. You can look at it and you can see how much is in one, how much the diversity in one, the organic matter in one compared to all the others. You can also see a range of expression. Check out this. Using epifluorescence in ACR Orange, we essentially have a dye that glows when it's not absorbed over the whole slide, right? But when it's absorbed by life, it's held inside the life and only they glow. And so it makes the whole slide dark. So the more life, the darker the slide. The less life, the lighter the slide, right? And there's nuance, right? You can see the differences in color. But we just figured out, just with simple color coding, how to differentiate between different samples, how to differentiate their numeracy of life. And if you take a section that's really exemplary, has testate amoebae, has good bacteria, good fungi, all those things in there, it's going to be exemplary. And so you'll be able to compare that and really see, does this stack up? How does this compare to the other samples that I have? And so I value this so much because it's not just easier for the practitioner, the microscope operator, in review after they've taken the video, edited it so it's in a side-by-side. It's instantly easy, but it's also easy to show to other people. And so how do we do this in practice? It's very simple. We have a worst example for that region, for that soil type, and we have a best. And so, and it could be one of the best, it could be one of the worst. That's okay. This is also why this database is necessary, because we need those examples, we need those guides, because it's in relation to other things that we really see, and especially within the boundaries of your bioregion, what you're growing, your whether you're doing annuals, perennials, you know what I mean, ranching. All these things are going to be affecting what you're looking at, how it looks, how you're going to manage it, and in using this very clear, very clean, you see how much life there is. It really pairs well with a lot of common tests, and it makes it real because you actually can see your goal, you can see what you don't want, and you see where you are. And color. A child can do it. And numeracy, let's put 50 marbles here, 100 marbles here, 500 marbles here on tables. A child can tell you which is the largest, which is the smallest, right? And then in between. And so we can do that with our eyes instantly. It's counting instantly at a glance that we can't do as humans. And in fact, counting manually has all these problems. I've done videos on the limitations of bright field before. But one of the things I learned in my work, well, the reason I started writing this in the first place was a certified lab was given a sample that another certified lab was also given, the same sample, same soil, the same collection, and they got divergently different answers, and then it was exposed. And it was embarrassing, and they felt like shaken up by it, and so they wanted to figure this out, and so they assigned the task to me as their teacher, their permaculture teacher, and so I dove into it, and I had written regenerative soil, and I got this microscope, and I really started diving into all this, and I figured out you can't go from a drop to an acre by multiplying, because let's start off in a plane and say you're 5 degrees off. How off are you in 6 hours flight? You're nowhere near your destination. So we need to recognize that manual counting, human counting, not so good. for a variety of reasons. We're not so good at it. And we're really testing each other's ability to calculate and count against each other and the sine wave of expression. So whenever fungi appears, it's going to appear and it's going to start off from a piece of hyphae or a spore and spread out from there. So you're going to have relative abundance and relative absence of fungi depending on where you hit in those scenarios. And so it's really important to understand that everything is in the sine wave of expression. Even, pH changes 4 to 9 sometimes every micrometer in the soil. And that differentiation is great because it causes an exchange. When we shake things up and get an average pH, that's also helpful. But we have to recognize that it comes from an aggregate of that it comes from a larger range of expression. And so we're also not able to, as I said in other videos, we're not able to see all the bacteria. So there's bacteria that all light microscopy, no matter how good it is, 1000x resolution is the limit of resolution. After that, you're just zooming in on the same resolution. So 2000x Omax, not real. That's not real. That's actually just not real. You can try to do it, but you can't sharpen the image is the one way to put it. So the image only stays sharp up to 1000x, and then you can zoom in closer. You can make images bigger, but they don't have the ability to resolve and become sharp. So what we do know is past 1000x, smaller than any light microscope can see, there's bacteria. So you're never going to see it all. And in fact, if you get a really good machine like this at 1000x, you'll see things teasing at your vision. And you'll be like, what is that? And that's stuff that's smaller than this can see. Or right at the edge, right? And also those things swim up and out of your focus. So they like glimmer like they're almostlings. So that's the thing, is it's like we don't have the ability to count, but the dye does have the ability to dye them, and they can absorb it even if we can't see them, and the dye can disappear. So, that's my way around it. Okay? And plus a child can look at the color coding and know it too. You can co-create the learning moment and the decisions and the evaluation with your clients if you're a soil consultant, right? This is how we're going to get farmers, on board, because they'll be able to actually see and verify these things. So these two labs, they decided that they were going to do it in matching their arms, measuring together, and they were still wrong. And so the numbers multiplying up to scale solutions doesn't work. I started doing things by ratio because in biology, you're going to do things by ratio so it can scale and compare properly. Because, the wolves to deer, you're going to do a ratio because you're always taking a sampling. You can't catch all the deer, all the wolves. Same thing with fish. You're going to take a sampling and then do the ratios for that sampling. But there's no way to be like, we catch all the fish and all the predators and count them all and then we'll know. That's insanity. So we can't do that. So we can't multiply either. But ratios stay true, and they keep the margin of error, the original margin of error. We don't exaggerate it. Because, again, humans aren't that great. We do flow cytometry that's better. Now they're doing flow cytometry with AI that's improving things. But flow cytometry doesn't work with soil solution, right? Things are too big. And you kill things and add chemicals to it for the flow cytometry. So we have the best of all worlds by simplifying with this side-by-side methodology. Because there's also things like colonies and organic matter where the bacteria is all over it. You can't count that, but you can dye for it, right? You can have Archuleta Orange. And so I came up with this because I was trying to solve this problem. And to zoom out even further, all soil science that's numerically based, when they're multiplying, when they're calculating, to the gram or to the acre or what have you, it's all wrong. And that's simply because they take one of these test tubes I was doing some plant material in here, plant sap. That's why it's green. But they fill the soil up to here in it. I'll do a picture. And then they tamp it down so there's no air. And then they fill it up with water, or they fill it up with water and then add it to the next, like 1 ml worth. They displace it 1 ml and then add the water to the rest. The problem with all that, first of all, it's really hard to pour in exactly 1 ml. You tend to go over or under. But regardless of that, the amount of air is displaced by the water when you're doing it. So it's super efficient, or you're tamping it where it's less efficient. With compacted soil, it's sand and silt. You just pour it in and there's no need to tamp. There's no air. It's compacted, right? I have that soil. But if you've got really good garden, because I'm regenerating it, but and I've doubled my organic matter in a year, but hold on. With really good loamy soil, it's 50 to 60% air. All that crumb structure, it's airy, it's squishy, it's loamy. That is 50 to 60% air. So you're tamping it down, and you're reducing the area you're testing and taking numbers from by 50 to 60%. So this can be up to 50 to 60% larger spatial area of your soil, three-dimensional air space, than this compacted sand or silty or even compacted heavy clay soil. So the numbers between them are meaningless because they're multiplying, because they're trying to count and they're getting away from ratios. If it was in ratio, it'd be much more comparable, with the understanding that we're not great at counting, with the understanding that even with our great technology, it's okay at counting, and we can figure out that it's not that, because we do DNA testing as well. So the numbers are wrong. Maybe we should rethink the whole numbers thing and see things and show things in a way that people actually can, in their gut, be like, that one. Look at it. There's more life. Look at it. That's the darkest slide. That one. Which compost is that? You go out and you go check it out. You're like, this is obviously the best compost. Now that I look at it and compare it to the others, knowing that this is the good one, you can just see it. And then they'll start describing the attributes. This is more broken down. This is darker in color. This is holding more moisture. This, they may smell it, right, and say it smells better. That always makes me nervous, because unless you're like quite sure that there's no Aspergillus fumigatus and other things like that in there. So this is a way that I was able to solve all those problems by using Acridine Orange. Now, if you look it up, you're going to be like, oh my gosh, Acridine Orange. Like, there's a safety sheet for it, right? There is for, everything, that we're using in a lab in microbiology. That's part of the standards. It's a standard microbial and biological and medical stain. Acridine Orange is really incredible. It's used in malaria, fighting malaria. It's used for fighting cancer. It's also a coal tar derivative, so nature's used to this. So what happens is we use it so diluted that the microbes can eat it, and then they can, if we wait too long, it disappears. It gets remediated. So I knew in using this that this was going to be a good route. And also I had dye envy. So do you guys ever watch Journey into the Microcosmos on YouTube? I love them. I wanted those pictures, those glowing, amazing pictures, and figured out it was Acridine Orange. And so I was like, oh my gosh, I got to figure this out. And then when I learned that this was out of range, I was upset. And then I learned that, well, I was upset, and then was like, I don't care, I'm going to figure it out. And I ordered it. And then I started playing with it, and then figured out that everyone who made those things about how it's out of range was using eyepieces. It's out of range if you're not using a camera. If you're using a camera and you can adjust the brightness, like we can here, you can absolutely use Acridine Orange in a different range. So I was so excited about this. This was like a breakthrough. I was like, I'm finally able to get good pictures like all these other people and have it be alive. Because that's the thing, is what everyone else is doing, they're counting, they're measuring, but they're also using fluorescence diacetate. They're keeping it in their freezer. When they want to use it, they put it in the microwave, and then they combine the microbes with it. Yeah. So it's not in situ the life that we're just getting to view. It's a manufactured environment, and highly unnatural, highly artificial is actually the scientific word to describe that. And so I like having just 1 to 100 dilution or 1 to 10 dilution, depending on what we're doing. If you just want to look at the life, 1 to 10 is great. But if you want to put it through a hemocytometer and do the side-by-sides, 1 to 100 dilution, and then I just take 1 ml out of that, put it into a separate tube. And as you can see, I like working in small amounts. It's because of my DNA testing experience. I'm very used to tiny amounts being sufficient. So you do fractions of a drop, fractions and fractions and fractions of a drop with micro pipettes. So it's amazing. So 1 ml of the 1 to 100 from the other tube, that's compost or soil to water, distilled water. And then I add 2 drops of Acridine Orange that's been diluted twice, right? Right. And so there's so little waste. It can be absorbed by the biology right there and then, and you might have to add more drops to get it to sustain, for you to take your pictures and do your evaluation. And so, yeah, it's an incredible dye. I saw all these people with microscopes that were like 3 times as much money as mine using it, and I was like, I have to figure this out. Your images are too good. And so that's what I did, and it's a total game changer. It is, like I said at the beginning, it comes in a powder usually, and all powders are carcinogens because powders are hard to get out of the lungs. It's like talcum powder that's carcinogen. I mean, even balsamic vinegar and guitar pickups are carcinogens now. So keep this all in mind. Wear gloves when you handle Acridine Orange. If you're working with the powder while you're making things, or any powder, this is the same when you have water-based dyes that and this is non-toxic, but it's still, if that powder gets in your lungs, there's a problem, So wear a mask when you're doing it, if you're doing that, or get it already pre-mixed from a friend, because guess what? When you mix it, this is the double dilution, right? The single dilution is going to last me forever. So you get a friend with some single dilution, you get some from him or her, and then dilute that, and then you'll have tons. So I really use so little, it'll shock you if you actually do the work. The whole recipe, all the directions, all of this is inside this book, Regenerative Soil Microscopy. I wrote this book so that folks could literally just start in on this and do the work. This is the hydration. This is how to hydrate it and then dilute it, because you have to hydrate it, that powder, and then dilute it. And here's the side-by-sides in the book. There's the hemocytometer. So it's all in here, as well as to, how to calculate things, how to count things. It's all in there. So you can do it both ways and prove to yourself that it's better and more accurate to line things up and compare them than it is to measure and count. But I show you how to do it all, show you the history of it, so you understand how it all works in that book. Because I really want people to, because that's how I did it. I had to know. I'm like that child who they're like, that's the way it is. And I'm like, I'm going to go and open the door and see if that's true. Like, right? So I can't help it. I'm a citizen scientist. And because of that, I've had all these breakthroughs like manual lighting. Yeah, that's biochar right there. And so I just feel so much gratitude that I want to share this with the world so that you all know how this is done, why it's so much more powerful, and why you can do it and get involved. If you like this book, check out this book. Go to thepermaculturestudent.com to get this book signed with free U.S. shipping. And it's also on Barnes & Nobles, Amazon. You can ask your local library to order it, your local bookstore to order it. It's in all the catalogs that they would use and they would have access to. Then that goes globally too. It's in Australia. It's in Britain. It's all over. So enjoy that book. Enjoy Microscopy. I'm Matt Powers. Grow abundantly, learn daily, and live regeneratively. This is Regenerative Soil Microscopy, and it's something you can do too. Regenerative Soil Microscopy is the first soil microscopy book of its kind ever available. It focuses on the soil biology, compost, mycorrhizal fungi, rhizophagy, and so much more that is the life that makes all of our plants nutritious and healthy. And it's also a how to operate the microscope, how to do stains, the recipes, the directions, the step-by-step. That's all in there. But not only that, there's a morphological guide so you can compare what you're seeing against proven lab certified samples. So this is a game changer. This has never been offered before. If you want to see examples of the fungi and bacteria that matter, the protozoa, the nematodes, the macroarthropods, the microarthropods, all the soil microbes, all the trophic levels. If you want to see examples of them, you want to be able to compare them, you want to be able to look at the mouth parts, you want to look at a gallery of images so that you can sharpen your eyes so that you have a reference of comparison as you travel and explore soil, compost, indigenous microorganisms, even roots and plant leaves. We cover it all. And it goes beyond bright field. So it's dark field, it's epifluorescence lighting so you can see the invisible fungi, you can see the phosphorus where it goes and where it glows. And also manual lighting, which is a new creation that allows us to visualize roots and plant surfaces at a completely new level and allows us to see them in a much more natural and understandable light. If we don't look, we don't see. If we don't look through multiple lenses, we miss what is otherwise unseen. Mycorrhizal fungi is invisible in bright field microscopy. New methods, new interpretive keys, new insights, new discoveries, and next level results. We even have case studies comparing Johnson Sioux to hot composting, to vermicomposting, to dead sandy soil, to a compacted dirt road, effective microbes comparisons to EM1, EM3, to brews of purple non-sulfur bacteria, and even collections of indigenous microorganisms. We look at them all and evaluate them all together in this book. There's case studies, there's how-to, learn how to do the microscope, but also there's morphological guides, there's sizing guides. There is so much information in this book that has never been available ever in one place. So compare the images to the tests and see for yourself. Get all the data, all the images, all the sample types, all in one book. Order your copy today at thepermaculturestudent.com. Thank you so much for being interested in soil and soil microscopy, because this is the only way that we're going to really know if our soils are regenerative. Regenerative soils are the linchpin to healthy plants and to healthy people and to healthy futures. Thank you for being part of this great arc of regenerative soil. I'm Matt Powers. Grow abundantly, learn daily, and live regeneratively. Click the link and look inside the book. Check it out. You won't be disappointed. I guarantee it. You won't be disappointed. Check it out. I'm so excited. Have a great one, guys. And check out the